In this talk, we will learn about the different features of the organisms that come under the domain of Archaea. It is estimated that life on Earth evolved around 3.5 to 4 billions of years ago. The first living organisms that evolved were prokaryotes. The word prokaryote comes from two words: pro, that means before, and karyon, which means nucleus. Thus, prokaryotes are organisms that were there before the evolution of a nucleus. All prokaryotes are single-cell organisms, and they can be found in one of two domains. They are present in either the domain of bacteria or in the domain of Archaea. The word bacteria means a short rod or a cane. It is indicative of the appearance of the bacteria that the early researchers observed under a microscope. Archaea, on the other hand, means ancient or primitive. Thus, these organisms are considered to be older than bacteria. All prokaryote cells do not have membrane-bound organelles, and thus the cell composition is quite simple compared to eukaryotic cells, which are much more complex due to the presence of numerous types of membrane-bound organelles. All prokaryotes can be grouped into one of two kingdoms. The first kingdom we're going to look at is Archaebacteria. This comes under the domain of Archaea. Now, Archaebacteria comes from the word archaea, which means primitive or ancient. And thus, these are primitive or ancient bacteria. They are considered to be the ancestors of eukaryotic cells. Prokaryotes can also belong to the kingdom of eubacteria. The term eu stands for true, and hence the organisms that belong to this kingdom can be roughly translated as the true bacteria, which essentially states that they are not archaebacteria. Archaebacteria, or Archaea as they are known as nowadays, are single-celled prokaryotes. They are considered as microorganisms because they are quite small and can be observed under a microscope. They were originally identified in extreme environments and hence were called as extremophiles. However, we now know that they are more widespread than we originally thought, and they can be found in many diverse environments. Archaea are a lot harder to grow in laboratories compared to bacteria, and hence the information that we have about Archaea is a lot less compared to what we know about bacteria. Archaea come in a variety of shapes and sizes. Some are extremely small, being around 300 nanometers, while others are quite large and can be up to 10 micrometers. They also come in a variety of sizes, where some of them are round, as we can see in the figure. Some others are rod-shaped. We can also have some that are square-shaped and extremely flat. We can also have others that have irregular shapes. The archaeal cell is quite similar to bacterial cells, and hence, for a while, archaea were considered to be different types of bacteria. But we now know that they are quite different from bacteria as well. Now, some similarities with the bacterial cell include that they lack membrane-bound organelles because they are prokaryotes. Some of them may possess a glycocalyx, which is usually more abundantly found in bacteria, but in the case of the archaeal cell, it is not commonly present. Some archaeal cells have flagella, which aids in motility and movement. The archaeal flagella is structurally much different than the bacterial flagella, and hence it has its own name, which is the archillum. In fact, the archillum is actually more similar to bacterial type 4 pili than it is to bacterial flagella, and hence it is thought that the archillum and the bacterial flagellum show signs of convergent evolution. Talking about pili, the archaeal cell does show the presence of pili as well as fimbriae. These cell structures do not aid in motility or movement, but they do aid in other features such as attaching to surfaces or adhering to other cells. They also help the archaeal cells to be part of biofilms. In addition to fimbriae and pili, the archaeal cell shows the presence of other structures that are not seen in bacteria. These structures include the cannulae and the hemi. The cannulae are rod-like structures, while the hemi is a hook-like structure, and both aid in promoting attachment of the cell to different types of substrates. A cell wall is commonly observed in archaeal cells. However, the cell wall is different in composition from bacterial cells because it lacks peptidoglycan. Most archaeal cell walls are made up of glycoproteins and glycoconjugates. In the case of some archaea, they may have a cell wall that has pseudopeptidoglycan or pseudomurine, which is similar to peptidoglycan. Some archaeal cell walls have methanochondroitin instead of pseudomurine or pseudopeptidoglycan. This is also a polymeric substance and is similar to a component found in the cartilage of animals. Many archaea possess a surface layer or S layer on the outer part of the plasma membrane. These S layers are considered to be part of the cell wall and are made up of glycoproteins. They come in many shapes and forms. The S layers can be made up of glycoproteins that are embedded completely into the plasma membrane, or these glycoproteins can be associated with the plasma membrane but not completely embedded. In the case of some archaea, the S layer proteins are present on top of a polymeric cell wall that is there. Many archaeal S layer proteins are glycosylated, and this was a seminal finding because for a long time it was thought that the process of glycosylation, which is the addition of sugars to a protein, was observed only in eukaryotes. The S-layer proteins cover the entire cell and can form these two-dimensional paracrystalline structures. When we look at the cells under an electron microscope, we can see dazzling arrays being formed by these S-layer proteins. Like all living organisms, the archaeal cell has a plasma membrane. However, there are some differences in the plasma membrane of archaea and bacteria, even though both of them are prokaryotes. Let us first look at some similarities. In both cases, the plasma membrane is made up of phospholipids. The phospholipids have a glycerol backbone, and one of the hydroxyl groups of this glycerol backbone has a phosphate group attached to it. However, when we look at the lipid portion, in the case of archaea, it is made up of these branched hydrocarbons called as isoprenoids. This is different than the lipid portion of bacteria, or in fact eukaryotes, because they're made up of fatty acids. Another difference is the linkage of the isoprenoid group to the glycerol backbone, and it involves ether linkages. Whereas in the case of bacteria, the fatty acids are joined to the glycerol backbone with ester linkages. This ester linkage is also seen in eukaryotic plasma membranes. Thus, the plasma membrane of archaea is different than what is observed in bacteria and eukaryotes. In some archaea, the isoprenoids or the lipid portion can be joined to a glycerol backbone on both sides. When this happens, it can create a plasma membrane that is a monolayer. This is different than what is seen in bacteria and in eukaryotes, where we always observe lipid bilayers that make the plasma membrane. Archaeal cells are prokaryotes and hence they do not have nuclei. The region of the archaeal cell where we can observe the circular chromosomal DNA is called as the nucleoid or the nuclear region. Similar to bacteria, archaea can also have extrachromosomal DNA that is also circular. These DNA molecules are called as plasmids and are not essential for survival. However, the single chromosomal DNA that is present is essential for survival and growth. Similar to bacteria and eukaryotes, archaea have ribosomes, which are non-membrane bound organelles that play important roles in protein synthesis. The ribosomes of archaea are made up of two subunits. Each subunit is made up of ribosomal RNA and proteins. The smaller subunit is called as the 30S subunit. It has the 16S ribosomal RNA, while the larger subunit is called as the 50S subunit, which has the 23S ribosomal RNA and certain other small types of ribosomal RNA. In the presence of messenger RNA, these subunits can come together to form the intact 70S ribosome. This size of a 70S ribosome is also observed in bacteria, and hence it is considered as the prokaryotic ribosome. This size is different than the eukaryotic ribosome, which is slightly larger and is called as the 80S ribosome. It should be noted that even though the size of the archaeal ribosome is similar to that of the bacterial ribosome, the ribosomal RNA sequences that are present in the archaeal cell are much closer to eukaryotes than to bacteria. Archaeal cells show different modes of reproduction, like binary fission, budding, or fragmentation. Let us first look at binary fission, which is a mode of reproduction commonly seen in bacteria as well. It starts off with the parent cell that replicates its DNA to form two DNA molecules. Now, each DNA molecule anchors to opposite ends of the parent cell. Then in the middle, a septum is formed, which is a constriction, and this constriction deepens and deepens until the parent cell divides to form two daughter cells. Now, in each daughter cell, there is only one chromosomal DNA. And that chromosomal DNA is the same chromosomal DNA that was there in the parent. Thus, the daughter cells had the same genetic information as the parent. Thus, binary fission is an asexual mode of reproduction. Budding is another form of asexual reproduction that is also observed in yeast. In the case of budding, a small outgrowth is formed from a parent cell. This outgrowth is called as a bud that then starts growing in size. The cell then replicates its DNA, and one copy of that DNA is provided and packaged into the bud. Once the bud gains a certain size, it then falls off or breaks off or separates from the original parent cell. Thus, at the end, we end up with two cells that have identical genetic information. In this scenario, however, one cell tends to be smaller than the other cell. The smaller cell is the cell that formed the bud. Archaeal cells can obtain genetic information in non-reproductive ways. One such way is through transformation. Consider a scenario where an archaeal cell has died and its DNA is being released into the environment. This released DNA can then be taken by another archaeal cell through the process of transformation. Now, if this DNA was a plasmid, it could remain as a plasmid in the archaeal cell too, or if it's a DNA fragment, it can get incorporated into the genome of that archaeal cell. Another type of horizontal gene transfer is transduction. In this scenario, a virus is able to inject its genetic information into the archaeal cell, and that genetic information of the virus can be incorporated into the genome. Scientists have found different types of archaeal virus with many different shapes and sizes. Thus, these archaeal viruses can play important roles in transduction. Conjugation is another mode of horizontal gene transfer, where one archaeal cell can transfer its genetic information into another archaeal cell through cell-to-cell contact. Thus, archaea have different ways of obtaining genetic material in a non-reproductive manner. All organisms need an energy source, carbon source, as well as electron source in order to survive and grow. Energy sources are required so that the cell can synthesize ATP in order to fulfill the energy requirements of the cell. Now, this energy source can be a chemical compound, and those organisms that oxidize chemical molecules to obtain energy are called chemotrophs, while those organisms that obtain energy from light are called phototrophs. Carbon can also be obtained in different ways. Autotrophs are those organisms that will use inorganic carbon dioxide as the carbon source to make other types of carbon compounds needed in the cell, while heterotrophs are those organisms that will use organic molecules as a carbon source to make other carbon compounds. The electron source can be in the form of organic compounds or minerals or inorganic compounds. By taking into account the primary energy source, electron donor, and carbon source, different organisms can be classified into appropriate groups. In the case of Archaea, we have found photoorganoautotrophic Archaea, which use sunlight as an energy source, organic compounds as electron donors, and carbon dioxide as a carbon source. There are also some archaea that are chemoorganoheterotrophs. These archaea use organic compounds in order to obtain energy as well as electrons. They also use organic compounds as a carbon source. Some archaea are chemoorganoautotrophs, which are going to use organic compounds to obtain energy as well as electrons. However, they can use carbon dioxide in order to synthesize their organic carbon-bearing compounds. We've also observed chemolithoautotrophic archaea, which are archaea that are able to obtain energy from breaking carbon compounds and can obtain electrons from inorganic compounds. They are able to make carbon compounds using carbon dioxide present in the environment. Thus, we are able to see different types of archaea using different energy sources, electron donors, as well as carbon sources. For a long time, it took a while to classify archaea because archaea are difficult to grow in normal laboratory conditions. In many instances, they need specialized conditions to grow, or in some instances, they just grow really slowly. Thus, classifying them based on genetic information has been quite difficult using traditional methods. However, the invention of metagenomic techniques has allowed the classification of many different archaea. Metagenomics is a very powerful technique that is being used to identify organisms without any need of growing or culturing them. Thus, we can use any sample and determine what types of organisms are present in there. The technique involves obtaining a sample and extracting the DNA. The DNA that is extracted includes the DNA of all organisms that are present in the sample. And so, if we had archaea present in there, their DNA would also be extracted. The DNA is then fragmented and sent for sequencing purposes. Once the sequences of DNA are obtained, they are then assembled, and by using different types of algorithms, they are annotated and identified. These sequences can then be used for phylogenetic analysis. With the help of metagenomics, we have a lot of genetic information on different archaea, and thus the archaeophylogeny is being studied intensely. Since this is a rapidly growing field, there are revisions that happen periodically, and hence the nomenclature that we see today may be different in the future. Based on the information that we have today, the main superphyla that are present in the kingdom of Archaeobacteria are Dipan, Euryarchaeota, Tak, and Asgard. The superphylum of Dipan is an acronym for the phyla Diaphoretrides, Parvarchaeota, Enigmaarchaeota, Nanoheloarchaeota, and Nanoarchaeota. Some of the organisms that are present in these phyla have small cell sizes as well as genomes. Now, some are free-living, while others are symbionts, which is they have a mutualistic relationship with other types of archaea. Euryarchaeota is a superphylum that has members found in diverse environments. It includes thermophiles, which are organisms that live in high temperatures, halophiles, which are organisms that live in the presence of high salt concentrations, and methanogens. Methanogens are organisms that generate methane through anaerobic processes. All such organisms that are present so far have been identified as archaea. The Tak superphylum is an acronym for the phyla Thaumaarchaeota, Eigerarchaeota, Cranarchaeota, and Coraarchaeota. These include ammonia-oxidizing members, and currently the names of the different phyla are actually being revised. The superphylum Asgard is a more recently created superphylum and includes the phyla Lokiarchaeota, Thorarchaeota, Odinarchaeota, and Heimdallarchaeota. One fascinating aspect about this superphylum is that some of its members show the presence of eukaryotic signature proteins like actin, tubulin, small GTPases, etc., which are usually not observed in bacteria. This gives support to the idea that the first eukaryote may have evolved from an Asgard member. With this, we come to the end of our talk, where we learned about different features of prokaryotes and concentrated on the archaeal cell structure. We then learned about the different modes of reproduction observed in archaeal cells, as well as mechanisms of horizontal gene transfer. We then saw the classification of archaea and the different phylogenetic relationships among different archaeal superphyla.